Hi everyone, it's Miss Lopez. Welcome to your Chapter 5 notes on passive transport. In the previous video, we talked about the structure of the biological membranes. In this video, we're going to focus on passive transport, which requires no input of energy. First, let's discuss the function of the biological membrane, and that is to be selectively permeable. Biological membranes allow some substances, but not others, to pass. In other words, it separates the intracellular environment from the extracellular environment. There are two processes of transport across the membranes. The first is passive transport, which does not require metabolic energy. A substance will always move down its concentration gradient. In this video, we are going to focus on just passive transport. Passive transport can occur by simple diffusion through the phospholipid bilayer or facilitated diffusion, which is a type of transport, through channel proteins or aided by carrier proteins. Let's start with diffusion. Diffusion is the process of random movement towards equilibrium. It is a net movement from regions of greater concentrations to regions of lesser concentration. So what you're seeing in this diagram here is that the drop of ink may start as a single drop in this beaker, but over time it begins to spread until there is no net movement of molecules. Now that doesn't mean that the molecules have stopped. It just means that there is no net movement. Simple diffusion depends on three factors. The diameter of the molecules. So keep in mind that the smaller the molecules, the faster they will diffuse. The temperature of the solution. Keep in mind that the higher the temperature leads to faster diffusion. And the concentration gradient. So the greater the concentration gradient, the faster a substance will diffuse. In other words, let's compare two different beakers. In this diagram, the two beakers are separated by a semi-permeable membrane. Now let's look at the concentration gradient between the two. I've written percentages for each side of the beaker. And what you'll notice for this first beaker is that the concentration, 40% solution, is close to the concentration to the other side. So we are going to see a net movement of particles from this side to this side. However, because the concentrations are pretty close together, there will be the rate of diffusion will not be as quick as you see in the other example. So in the other example, you can see that there is a 10% solution on this side and a 90% solution on this side. Because the difference in the concentration gradient is so high, you will see faster movement from this side to this side in this beaker than you would see in this beaker. And that is what is meant by a difference in the concentration gradient. It's important to remember that the cell cytoplasm is an aqueous environment, as is the surrounding environment of the cell. Diffusion of each solute depends only on its own concentration. So if we were comparing sodium, we would only look at sodium. We would not take into account other molecules at this point. A higher concentration inside the cell causes the solute to diffuse out. Higher concentration outside the cell causes the solute to diffuse in. And in both these cases, they must cross the plasma membrane. Some molecules cross the phospholipid bilayer by simple diffusion. Some examples include oxygen, carbon dioxide, and other small nonpolar lipid-soluble molecules. This is a very important concept to remember. Again, molecules that are small, nonpolar, and lipid-soluble are able to cross the membrane by simple diffusion. Polar or hydrophilic molecules do not pass through, and they are not soluble because they are not soluble in the hydrophobic interior of the membrane. This might include amino acids, sugars, ions, and water. So the important thing to remember about this is that it's the hydrophobic interiors, in other words, the tails of the phospholipids that will prevent polar molecules from passing through. So I've drawn here a cross-section of the plasma membrane, and I'm going to remind you guys of the different parts of the phospholipid. First is the hydrophilic heads, which are polar, therefore interact with the aqueous environment. The other part are the tails. They are hydrophobic and will point towards the inside of the membrane and are not interacting with water. It is those tails that will prevent polar substances from crossing the membrane. So this area right here, the hydrophobic tails, will prevent polar molecules from passing through the membrane. Large molecules will also not be able to cross the membrane because they will disrupt or break the membrane apart, and that is not energetically favorable. So let's summarize the process of diffusion and facilitated diffusion. In this model of the plasma membrane, you can see that we have the tails pointed towards the interior of the membrane. Substances that are going to diffuse across the membrane passively include small nonpolar substances. The most easy example to remember are your gases, such as oxygen and carbon dioxide. And these molecules are not blocked by the hydrophobic interior of the membrane. For passive diffusion, substances will move from high concentration to low concentration. Another important factor of diffusion is that this process does not require metabolic energy. The other type of passive transport is called facilitated diffusion. Now, it's important that you guys understand with passive, there is still no energy requirement needed. In other words, we're not going to be using ATP. Facilitated diffusion, or more specifically, facilitated transport, is for small or large polar molecules. Substances such as water, amino acids, and glucose are examples. If the molecule is water, it is very important that we make note that the name of the channel that's going to allow water pass through is aquaporin. A channel is a transmembrane protein that acts as a bridge and allows the molecules to pass through the membrane without having to interfere or interact with the hydrophobic core. Molecules will still move from high concentration to low. With facilitated transport, because of the structure of the molecule passing through, that channel is required. It's important to be able to predict whether or not substances will passively move through the membrane or if they will require facilitated transport with the aid of the protein channel. It's also important to know that the protein channel is specific, meaning that only certain molecules will be able to pass through each type of channel. That's it for your notes on passive diffusion. Thanks so much for watching.